Hello and welcome back to the Computational Genomics Lab. Today we are going to go over what data are we going to study? What are we going to analyze? How are we going to learn all this bioinformatics? Well, I'm really excited to get into it. And basically it's around a drug called doxycycline. And we have lots of data, unpublished data. I'm hoping maybe we can publish a paper together by learning bioinformatics skills with a real-world application and how to make results and figures and do all the things we would do in a research lab. So let's get into it. And so what is doxycycline and why would I even care? Well, it's a prominent drug that's used all over the world. It's an antibiotic and it's also used to cure acne. And so it's doing something pretty remarkable in the human body. And the question is, how does it do that? What is the molecular aspects that the dox is doing, doxycycline, I'm just going to call it dox, doing to make acne go away and kill bacteria? And it's surprisingly unexplored. There is some work on it, but really in the molecular biology science experimental world, what dox is used for is to induce a gene of interest. And that's sort of shown here, where you have this protein RTTA that likes to bind to dox, but until it binds to dox, it just floats around. It's just doing things, and it only recognizes this TRE promoter here. And so you can put in your favorite gene, I'm calling it gene X here, and by adding dox to the cellular media, it will induce RTTA to go turn on your favorite gene, and then you can track it through time. Let's go over what all that sort of means, and we'll go stepwise here. And so you would add dox just to your media, just pipette in a little bit, and dox will then bind to the RTTA protein, change its conformation so that it becomes active, and goes to the TRE promoter and turns on your gene. So now by adding dox, you've only turned on just that gene you've placed in this system here. And so then you have a temporal understanding of when you added the dox, and you can do RNA-seq through time or other approaches to understand what are the molecular changes that occur upon exposure of this gene. And so that sort of looks like this. And you would do a control system where you would not add dox and follow the same thing. And that's usually unexplored. People just use it as a control, but that's what we're going to dig into because I think it could be very interesting. And so if you add dox, this is this gene X here on the y-axis saying what genes, this is just one gene, a plot of one gene that gene X is regulating. And you can see over time of it, after RTTA has turned this gene on, another gene turns on. And so we would, by association, say, well, gene X probably regulates this other gene, and it does so in a temporal manner. And that's kind of what we're going to be doing to analyze our data. Whereas without dox, this gene does not go up. And so that says that it had to be RTTA turning on gene X subsequently turned on this plot we're looking at here on the right, and through time, that target gene of gene X turned on. So that way you can sort of connect molecular circuitry of what is gene X regulating and when is it regulating it. Is it regulating it early, later in time? And so this is a temporally controlled induction system that's really important. But underneath this is something kind of scary. Uh-oh, dox regulates genes on its own. So if you didn't have RTTA in there and you just added dox, we discovered recently lots of genes change. Now the good news is they don't change until about 2 to 4 hours. But in this plot here on the x-axis, we have time of 12, 24, 48, and 96 hours. So basically no dox to 4 days. And what you can see is about 1,000 genes going up over time and 1,000 genes going down over time. And this has nothing to do with gene X, but it does have to do with dox. And that's really cool that we can use this control data that we have tons of. And that's going to be one of the premises for this analyzing this class and learning RNA-seq is to use this data and ask, what are these genes? What could there be their biological relevance in acne or antibiotic responses? And so this is important, and it can guide us to a scientific journey to say, what does dox do? When and where does dox regulate genes? And that's where we're going next. Okay, so yes, this is important, has real world applications, and we want to learn bioinformatics skills, so we might as well learn on something we can explore and use our scientific curiosity, ask new questions, and work together towards understanding this. Cool. So let's see how dox does these two phenomenal processes of antibiotics and acne cures. So RNA-seq will give us the answers. It'll tell us on a molecular level what genes is dox changing and when does it regulate those genes. And so we're going to take it a step further and do this in human and mouse and find out what genes are the same in both of these species. That way they might be a little bit more important. We'll do each species on its own so we can really hone our RNA-seq and informatics skills here. And we're going to learn a lot about the genome as we go too in different genes. We have this system built into both these cell lines where we can add dox. We already have the data generated, it's ready to go. You can download it in the link below if you want to start getting on it. And we put in, instead of GeneX, GFP. So this cassette is working, but GFP shouldn't really do anything to cells. So we're really just looking at what dox is doing on its own and study the pathway of this important drug. And these are in stem cells, so they're relevant stem cell system to use that we can compare mouse and human, which is another great bioinformatics skill to have, is, you know, comparing genes that are called conserved, or they do the same thing in two different species, means they're probably more core to the process of how a drug is working. And so here's just an overview of the data we have. So we have human and mouse stem cells with this construct in it. And we have data in triplicate, so there's 3 replicates, so we're going to learn some statistics as we go. How do you deal with replicates? Why do you need replicates, etc. So we have a 0 time point without dox, 12 hours, 24 hours, 48, and 96 hours of exposure to dox. So this will allow us to look and investigate what is this drug doing to stem cells. And we can start to see that in human. We have the same data set for mouse. And we also have more data that I'll bring in later in class, but this is just sort of an overview of where we're going to start. But we have data from earlier time points as well, so we can see what the most immediate effects of dox are. And so what this will allow us to do is ask the question, how does dox regulate the transcriptome, or all the genes in the genome, upon exposure to it? And this will be a great way to learn RNA-seq, all the statistics and best practices involved with it. And to have a real-world application is, I think, always a better way to learn. And we might find something really cool. So I'm very excited to see what we can all work together and find. And as a bonus, we have this same data set with ATAC-seq. And what ATAC-seq is it tells you where DNA has opened up upon dox exposure. And so we can start to learn what DNA is opening up, and then if the DNA opens up, does that gene turn on? Or really just generally, how does dox affect the epigenome, or the epigenetics of the entire genome? So we have 2 great real-world applications, and by the time we analyze all this data, I'm hoping you'll feel comfortable to analyze any data set. We're going to use pipelines that are available for any type, clip-seq, any type of seq you can think of. And the same practices we're going to learn for RNA-seq, and the statistical analysis of it will be applicable to anything you might need to do in your own research. Okay, so what will we learn doing all this? Well, I think the most important thing we're going to learn is practical bioinformatics skills. You're not going to just do some coding to do something that you would never do again. You're going to be doing something that might likely happen in your actual research career, where you get a time course of data, time course of a gene knockout, other things. That time dynamics are kind of a tricky thing to deal with in data analysis, and we're going to do lots of that. And so it's really a practical bioinformatics skills with a real-world application, and I hope that'll build intuitive knowledge base that allow you to feel comfortable to move on and apply these skills elsewhere in your own research, and give you some confidence to do that. We're going to learn about genome-wide transcription and epigenetics, one of my favorite topics. We're going to see the whole genome changing and responding to this drug, and that will be exciting, and learn how and when and where this drug is working. And we're going to make great figures. I love figures. We're going to learn all the code for how do you make nice figures. Again, real world application, things like heat maps are used all the time. Meta plots, we're going to go over that, where you sort of average all the data across the genome. Basic plots, line plots, bar plots, all that stuff will be involved. And I think all those skills are used all across any informatics science. So that will be another real world application. And really, we're asking kind of a real research question is how does dox regulate the transcriptome and epigenome, and using time to follow that? What are the sequential steps in this process? And so I think it's possible. We've done this once before, but I think because this data is unpublished, but it's free for the whole world and the students in the class, could we actually publish a paper from this? Well, we're going to learn new scientific insights. We're going to learn how to make great figures. We could start to learn how to put these results together. We're going to have markdown of all of our code, so it's going to be 100% reproducible. Those are the main ingredients for writing a research paper, and it would be so cool to have all the students be authors on a paper and invite the whole world through our YouTube, GitHub, and class website to take these classes and ask their own questions and submit data back or figures. And so it's really kind of, my hope is it can be a citizen science project where we can work as a collective, all with free resources, all with free data, to be 100% reproducible, find some cool insights, and write a paper together. I'm motivated, and I'm ready to do everything I can to accomplish that goal. And so I think with that, I think you've got this. We can do this. We've broken it down into small steps and bits. Just work through them one step at a time. And let's go. Let's do this. Let's find out what dox does in human and mouse stem cells, and hopefully write a paper with beautiful figures, great results, and new insights to contribute to the scientific body. Okay, with that, we'll see you on how to run the RNA-seq pipeline next.